Yes, to this presentation of the review questions for Form 6. You can also download this from St. Joseph University in Tanzania platform. We have prepared some questions, some biology questions for paper 1 that you can revise them the time that you are preparing before the national exam. We have some questions that we have prepared here, and these questions, if you starting with the paper 1, we have the first question, the first review question, which this question is a reproduction question, and it has 10 marks if it is in section A. So question number 1A is saying the number of complete set of chromosomes into bracket ploid in a geminoepithelial cell of human being is 46. Calculate the number of ploid in the following. If you are given a question like this, as I've already said, that this question is in reproduction, and it has, if it is in paper 1, section A, it is in paper 1, and it is, if it is in section A, then it has 10 marks. If I given a question like that the number of ploid, the number of complete set of chromosomes in a the number of complete sets of chromosomes in a geminoepithelial cell of a human being is 46, then we know that the number of chromosomes in the geminoepithelial cell, because the geminoepithelial cells are diploid, then the number of chromosomes, that means 2n is equal to 46. 2n is equal to 46 because the geminoepithelial cells are diploid. So if you are given, you are told to calculate the number of chromosomes in different cells from there, then because you know 2n is equal to 46 because the geminoepithelial, then if we divide 2 and divide by 2 both sides, then n is equal to 23. N is equal to 23. Then after you have the value of n, the questions from this, the question is saying calculate the number of ploid in the following. Roman 1, spermatogonia. Spermatogonia. Roman 1, spermatogonia. It's A, Roman 1, spermatogonia. Spermatogonia. Spermatogonia are the cells that these spermatogonia are diploid cells. And if these cells are diploid, because these cells are diploid, that means spermatogonia are 2n. Now because n is equal to 23, then because this 2n is equal to 2 times 23, which is 46. So the number of chromosomes in the spermatogonia is 46. That's Roman 1. Roman 2, the number of chromosomes in the primary spermatocytes. The transformation of spermatogonia to spermatocytes involves mitosis. Therefore, the primary spermatocytes are also diploid. The primary spermatocytes are also diploid. Primary spermatocytes are also the primary spermatocytes. The primary spermatocytes are also diploid. If they are diploid, that means 2n. Therefore, if n from this, if n is equal to 23, then 2n in this case will be 2 times 23. The answer is 46. So the number of chromosomes in the primary spermatocytes is also 46. Roman 3, the number of chromosomes in the secondary spermatocyte. Roman 3, secondary spermatocyte. Secondary spermatocyte. This is Roman 3. Secondary spermatocyte. Secondary spermatocytes. Spermatocytes. Secondary spermatocyte. Each secondary spermatocyte this word here is continuous with no space. Spermatocyte. There is no space. So each secondary spermatocyte is haploid. These secondary spermatocytes, these are haploid because they result from meiosis. Each is haploid. Therefore, if each secondary spermatocyte is haploid, that means the number of chromosomes is N. Therefore, here, if N is equal to 23, then the number of chromosomes in the secondary spermatocyte is 23. And the last one, Roman 4, is spermatids. Spermatids, the number of chromosomes in spermatids. Calculate the number of chromosomes in spermatids. This is Roman 4. Spermatids, again, the spermatids are these results from second meiotic division, which the second meiotic division maintains the haploid number of chromosomes produced in the meiotic one. Therefore, spermatids are also haploid. The spermatids are haploid. That means N is equal to 1. Therefore, even the number of chromosomes here is 23 because the spermatids are haploid. That's A with the 4 Romans. And in this question, when solving this question, the first showing this is 1 mark. Then each calculation of these 4, each of them has 1 mark, making a total of 5 marks. That is part A of the first question. Part B of this question is saying the trophoblastic cells secrete hormone. I mean, the part B of this question is saying the trophoblastic cells and placenta play role as endocrine gland in pregnant women. Explained by giving 5 points. Trophoblastic cells and placenta. Trophoblastic cells and placenta. Trophoblastic cells and placenta play role as endocrine gland in pregnant women. Explained by using 5 points. Trophoblastic cells and placenta play role as endocrine gland in pregnant women. Explain using 5 points. That's the part B of the first question. Then in this question, it's also found is of reproduction, and it is this. In this case, is part B of the first question. And this question, from this, the question demands the student to write the endocrine roles of the trophoblastic cells and the endocrine gland. The trophoblastic cells secrete a hormone. First, these trophoblastic cells are the cells, these trophoblastic cells, these are the cells of the trophoblast. The trophoblast, these are the cells of the zygote. Usually the cells of the zygote. So the trophoblastic cells secrete a hormone called the human chorionic gonadotrophin, or otherwise abbreviated as HCG. The trophoblastic cell, abbreviated as the trophoblastic cell, secretes a hormone called human chorionic gonadotrophin, or this is abbreviated as HCG. Sometimes it may be called chorionic gonadotrophin. and this will be abbreviated as CG. If it is chorionic gonadotrophin, then it is abbreviated as CG. If it is human chorionic gonadotrophin, then it is abbreviated as HCG. This HCG prevents the breakdown of the corpus luteum. Maintains the activity of the corpus luteum because it prevents the breakdown of the corpus luteum. It's known that the corpus luteum is an endocrine structure in the first 3 months of pregnancy because it secretes large amount of progesterone that maintain the lining of the uterus and small amount of estrogen that repairs the lining of the uterus called the endometrium. So HCG, like the HCG maintains the activity of the corpus luteum. That is the first point. And in this case, for the trophoblastic cell, only it has as an endocrine role is usually regarded as it has the endocrine role because of the secretion of that single hormone. But on the other hand, the placenta, as an endocrine structure, it secretes a number of hormones. It secretes 4 hormones. These hormones are estrogen, progesterone, human placental lactogen, and human chorionic gonadotrophin. Starting with the estrogen among the hormones which are secreted by the placenta. Estrogen facilitates the growth and repair of the lining of the uterus called the endometrium. So estrogen facilitates the growth and repair of the lining of the uterus as one among the hormones which is secreted by the placenta. The second hormone which is secreted by the placenta is progesterone. Progesterone is a hormone, a steroid hormone secreted by the placenta, and progesterone maintains the lining of the uterus. In other words, you can simply say that progesterone maintains the pregnancy because it maintains the lining of the uterus. The third hormone secreted by the placenta is human placental lactogen, abbreviated as HPL. HPL. This stands for human placental lactogen. This is abbreviated as HPL. The human placental lactogen stimulates growth and development of breasts. This is the preparation for lactation. Stimulates the growth and development of breasts, and this is in preparation for lactation. It's just the preparation for lactation after birth. The last hormone secreted by the placenta is the same as the hormone which is secreted by the trophoblastic cells of the zygote. This is human chorionic gonadotrophin, abbreviated as HCG. It has the same law that prevent the breakdown of the corpus luteum, and therefore it's a hormone that maintains the activity of the corpus luteum. And I've already said the corpus luteum is a yellow body which develops under the influence of a luteinizing hormone. That the luteinizing hormone converts the remains of the Graafian follicles into a yellow body called the corpus luteum. The corpus luteum is an endocrine structure in the first 3 months of pregnancy. This time is called the trimesters. So the corpus luteum is an endocrine structure as it secretes the progesterone and estrogen in the first 3 months of pregnancy called the trimesters. Yes, in this, so 1 hormone secreted by the trophoblastic cells of the zygote and 4 hormones secreted by the placenta makes up the 5 points as they were required in the question. Each of the point will have 1 mark, making a total of part B making a total of 5 marks. So the grand total in this question number 1 is 10 marks. Then the second question is a question of respiration. This question is, the second question has again 2 parts, A and B, and it's a question of respiration. Because it is in this section A, then it has also 10 marks. And this question, part A, says what are the laws of the mucus and surfactant secreted by the epithelial lining, the epithelial lining, the nasal passage and alveoli respectively. This question is in respiration, and because it is in this paper 1, section A of paper 1, then it has 10 marks. And the first, this question, part A of these 2 is the one that I have said. It says, what are the laws of the mucus and surfactant secreted by the epithelial lining, the nasal passage and alveoli respectively. The law of mucus and surfactant secreted by the epithelial lining the nasal passage and alveoli respectively. This question is let's start with the functions of the mucus first. Mucus, the functions of mucus in the nasal passage and the alveoli respectively. Functions of mucus. Mucus performs the following functions. The functions of the mucus in the nasal passage and the alveoli, in the parts of the nostril where the air passes, and the alveoli respectively. Mucus first traps dust and bacteria that enters with the air via the nostril. The mucus traps the dusts and bacteria that have managed to pass through the nostrils together with the air. Also, mucus moistens the air that passes down to the alveoli. The moisture is important because the gases, when they reach there in the alveoli, the gases diffuse more easily when they are in solution form. So the mucus moistens the air that passes down the alveoli. Those are the two functions of the mucus. Then the functions of the surfactant. Surfactant is a detergent-like chemical that starts to be secreted by the epithelial lining of the epithelial cells lining the nasal, the nasal passages and the alveoli. The surfactant, the alveoli, the lining of the alveoli or the wall of the alveoli consists of the cells that secrete a special detergent-like chemical called surfactant. Surfactant starts to be, in humans, surfactant starts to be secreted in the 23rd week of pregnancy. And in most cases, the babies that are born before reaching the 23rd week of pregnancy, then they're termed as premature babies because they are born they lack surfactant. Surfactant performs the following function. First, it lowers the surface tension lining the alveoli, the lining of the alveoli or the wall of the alveoli, hence reducing the energy required to breathe in and inflate the lungs. Also, surfactants kills bacteria trapped in the incoming air through the nostrils. But again, surfactant increases the rate of gaseous exchange in the lungs. Why? Because the surfactant also provides the moisture that dissolves the gases, and the respiratory gases diffuse more easily when they are in solution form. So the surfactant, by moistening and dissolving the gases, then it speeds up the rate of the exchange of carbon dioxide and oxygen across the lungs. That's part A of the question. And in this question, again, at least one function of mucus will have 1 mark, and 3 points of the function of surfactant, each 1 mark. So for the functions of the function of the surfactant makes up 3 marks. We'll have 3 marks plus 1 of the function of the mucus, making a total of part A, making 4 points of part A. Part B is again of this a question of the same topic, which is gaseous exchange and respiration. And this question says, explain the following biological facts. Roman 1. Number 2B, Roman 1, which is still respiration, but it's part B. Roman 1 is saying the initial stage of glycolysis involves the use of ATP. The initial stage of glycolysis involves the use of ATP. Yes, it's true that the initial stage of glycolysis uses ATP, and it's because the initial stage of glycolysis is phosphorylation of the sugar, or phosphorylation of glucose, where in this initial stage the glucose is phosphorylated. The glucose is phosphorylated, means the inorganic phosphate from ATP are added to the glucose to make it more reactive. So the phosphorylation occurs twice, because the first phosphorylation is in the conversion of the glucose into glucose 6-phosphate. The first phosphorylation is in the conversion of glucose into glucose 6-phosphate. Glucose 6-phosphate, and here there is an input of 1 ATP. Here this 1 ATP is used. 1 ATP is used where it produces only this ADP, because the ATP, the hydrolysis of ATP gives energy, ADP and the inorganic phosphate. The phosphate is the one that replaces the 1 hydrogen on the OH, on the first carbon of this glucose, and I think there is also hydrogen. So the glucose is phosphorylated to give glucose 6-phosphate that uses 1 ATP, and then the glucose 6-phosphate is isomerized to fructose 6-phosphate. Later on, the fructose 6-phosphate is further phosphorylated to form fructose 1,6 biphosphate. So in the first stage of glycolysis, uses 2 ATP, because phosphorylation occurs twice. First is in the conversion of glucose to glucose 6-phosphate, which later the glucose 6-phosphate is isomerized to form the fructose 6-phosphate. Later on, the fructose 6-phosphate is converted to fructose 1 is then phosphorylated to form fructose 1,6 biphosphate. So that's why the initial stage of glycolysis uses ATP. And the phosphorylation serves several purposes, because first, the phosphorylation of sugar increases the reactivity of oxygen in the phosphate ester bond, and therefore phosphorylation activates the sugar, makes the sugar more reactive. That means glucose 6-phosphate is more reactive than glucose. Secondly, the addition of inorganic phosphate to a glucose molecule make glucose 6-phosphate. This glucose 6-phosphate, it has the phosphate group. That means the glucose 6-phosphate is charged, and therefore this process traps the glucose within the cytoplasm, because once it is glucose 6-phosphate, it is charged. Therefore, it will be repelled by the phospholipid bilayer. Roman 2 of this question is saying carbohydrate produces less energy than lipids. It's about, we are told to describe these biological facts. Carbohydrates produces less energy than lipids. The carbohydrates, first, chemically, carbohydrates contain fewer number of hydrogen atoms than lipids, and therefore their oxidation produces fewer ATP, because if the carbohydrates contain fewer number of hydrogen atoms in their molecule, that means in the electron transport system, fewer pairs of hydrogen will be carried by the oxidized NAD to form reduced NAD. And we know in the electron transport system, the pairs of hydrogen enters in the electron in the chain and results into production of ATP. Carbohydrates produces less energy, or carbohydrates have lower calorific value than the lipids, because carbohydrates chemically contain fewer number of hydrogen atoms, and therefore this results in formation of fewer pairs of hydrogen that will be carried by the oxidized NAD entering the electron transport chain, and from this fact it results in production of fewer ATP. Roman 3, people residing at high altitude have dark red. Roman 3, people residing, this is a biological fact. We were told to explain the following biological facts. People residing at high altitude have dark red blood than their counterparts at the coast. Yes, this is because at high altitude, the people residing at high altitude have a high concentration of hemoglobin than their counterparts living at low altitudes, because at high altitude there is low partial pressure of oxygen. So the people residing at high altitude must have blood which is dark red, showing that they have high concentration of hemoglobin, which is a colored, red colored pigments responsible for binding with oxygen. So the people residing at high altitude have dark red blood because their blood is more concentrated with the hemoglobin than their counterparts living at low altitudes. This is an adaptation that enables the people to reside or to live at high altitude, because at high altitude, as you go up, the partial pressure of gases decreases, and in this case, the partial pressure of oxygen decreases with the altitude. So individuals residing at high altitude must have one among the adaptation that they have is their blood is dark red. That means the blood contains large number of hemoglobin responsible for binding with oxygen, even if the tension is very low or the partial pressure is very low. That is the end of the second question, the second review question. The third question is of principles. Yes, it is of principles of classification. Principles of classification. This question is again as like the other questions of paper of section A, paper 1. It has 10 marks, and part A of this question is saying, by using 4 points, explain why it is difficult to achieve the natural system of classification. It is difficult to achieve. By using 4 points, explain why it is difficult to achieve the natural system of classification. Natural system of classification is the classification system that considers both the internal and external features in putting organisms into their correct or their respective groups. It considers both the internal and external features in placing the organisms in their respective groups. Also, natural system of classification considers the evolutionary relationship among groups of organisms. Of course, the natural classification is the accurate type of classification, but it has never been achieved in the it is difficult to achieve the complete natural system of classification because of the following challenges. First, natural classification is a time-consuming process because using both the internal and external as well as the evolutionary relationship consume a lot of time. So natural classification is a time-consuming process. Therefore, because of this, it's difficult for a complete natural classification to be achieved. Also, it needs skilled personnel. That it's a classification system that needs some classification. It needs expertise. It cannot be done by everyone because it needs someone who is comfortable with or someone who has knowledge on both the internal and external features as well as the evolutionary features. Also, natural classification is difficult to achieve a complete natural system of classification because it is expensive in terms of cost and equipment, that it may require the taxonomist to move from one point to another in order to collect or in the act of gathering information. But also it needs the taxonomist. It requires the taxonomist to use some equipment, some special equipment when observing or when collecting the data. For that matter, it is expensive in terms of costs. It uses a lot. It needs a lot of money. It is expensive in terms of cost and or in terms of money, and it requires also some modified or some advanced equipment. Again, natural classification is not stable as it allows the addition of information and the discovery of new organisms. Natural classification changes with the advancement of science and technology. For that matter, it's very difficult for it to be achieved, for a complete natural classification to be achieved. But also, natural classification is tiresome and tedious because it involves the use of many features, both internal and external, as well as the evolutionary relationship, the evolutionary features. And sometimes the natural classification requires the taxonomist to trace the evolutionary background of the organisms, of which some of these evolutionary features, probably some of them cannot be observed externally. Then because of these facts, it's very difficult to achieve a complete natural classification. Part B of the question is again of the same topic, but this is in one of the subtopic of principles of classification, which is the dichotomous, the identification keys. Part B is saying you are provided with the following organisms. We have grasshopper, bee, centipede. We have grasshopper, bee, centipede, grasshopper, bee, centipede, spider, and millipede. Millipede. You are provided with the following organisms: grasshopper, bee, centipede, spider, and millipede. Use the following features in order to construct a simple numbered key. Use the following features in order. These features includes wings, the features. We have wings, and we are told to follow the features in order. Wings, hairy body, antenna. Hairy body, antenna, and hairy body, antenna, and legs. Then from this we're given this organisms. There are 5 organisms. 1, 2, 3, 4, 5, and we have 1, 2, 3, 4. We have 4 features. And then the question says construct. Use the following features in order to construct a simple numbered key. Wings. So we are supposed to construct a simple numbered key from the given organisms and the features. Then when answering this question, you first let's write. We are told to write the simple numbered key. This is the same as, is also called the bracketed key. This is also a bracketed key. This is a key in which the pairs of couplets starts at the same marginal level. That the pairs of statements, we know in the pairs of statements in this case starts at the same marginal level, and they are usually, each statement is followed by a dotted line and the number showing either the ending with if the number showing the next couplets that should be considered. So from this we have this. Let's start with the wings. For example, say 1A, organism with wings. Organisms with wings. This with wings here, dotted line, with wings, organisms with wings. In this case, we have how many organisms with wings from here? We have the ones with wings. We have this grasshopper. We have bee. Yes, these are the only 2 with wings. So with wings, there are only 2. Then you write, maybe you can either write go to 2 or simply 2 organisms with wings. Then 1B, organisms without wings. 1B, organisms without, the organism without wings. The ones without wings, that means this other. So the ones without wings, let's say go to 3. Number 2 is only for the ones with wings. So number 2A, only the 2 with wings. The next feature to consider is hairy body. So organisms with hairy body. Organisms, 2A is organism with hairy body. Organism with hairy body. And this with hairy body, this must be the B, because it's the one with hairy body. The one with hairy body. 2B is the organism without. Organism without hairy body. Without hairy body. And this, the one without hairy body, this must be grasshopper. Grasshopper. This is the one without hairy body. The ones without wings is number 3. So number 3A is the ones without wings. This one already identified and this already identified. So the next is antenna. Organism with antenna. Organism with antenna. The organism with antenna here. The organism with antenna is the ones with organism with antenna. Millipede has antenna and even centipede. This spider belongs to class Arachnida. So the arachnids lacks the antenna. So with antenna, that means organism with antenna to be go 2, 4. And 3D, which is the organisms. This will be for organisms. Organisms, this is organism with, organism without antenna. The organism without antenna, this is the spider, the organism without antenna. Then number 4 is for the organisms with antenna. There are 2, and we have centipede and millipede. Let's say the feature is legs. Then let's say organism, we can just differentiate them by using the number of legs per segment. So organism with 2 pairs of legs, organism with 2 pairs of legs per segment, of legs per segment. The organism with 2 pairs of legs per segment. This is the millipede, because the millipede is the one with 2 pairs of legs per segment. And 1B is organism without or with 1 pair. With 1, because these 2 are contrasting. This is 2, this is, so it's 2 pairs of legs, and this one will be the 1 pair of legs per segment. That means these are 2 states of a character that oppose one another. This one says they are contrasting because this shows that there are 2 pairs of legs and this one 1 pair of legs per segment. Per segment. And this is centipede. This is the centipede. So this is the simple number key for there were how many insects, there were how many organisms. They are not insects, they are organisms. And this, I think all of them belongs to phylum Arthropoda. So these 5 organisms, if you are given 5 organisms, then you must have 4 characters, of which each character will have 2 character states. One fits the situation and the other one is reject. And then if it is bracketed like this or simple numbered, it's usually the 2 states of a character starts at the same marginal level, followed by dotted lines, then followed by the number that indicates the next couplet that has to be considered in case you don't end with a single organism. So that marks the end of this this part B of question number 3. Part B of question number 3. Part C of question number 3 is a simple question. It's C of the same topic. So in this, here, there are 1, 2, 3, 4, 5, 6, 7, 8. That means each I have a mark, making a total of 4, plus the 4 marks in part A, it will be 8 marks. Part C is of the question that says, part C of question number 3C. A biologist found some new insects which were supposed to be placed in the taxa, among the tools which a biologist demanded for this work was a biological key. Why do you think a biologist needed a biological key? Give 1 point. This is again of this in the same a question in the same topic, which is principles of classification. And then if we are given a simple question like this, then if the biologist demanded a biological key for this work, then the importance of having the reason as to why the biologist needed this tool is for quick identification of an insect or of an organism, because the biological key, the identification keys are the biological tool for quick identification of an organism. So if the biologist this biologist found a new insect and then he demanded the biological key, it must be for quick identification of an organism. And that question has 2 marks, making a total of 10 marks in question number 3. The 4th question is a question of coordination, and it has, again, because it is in the same question, in the same section, it has 10 marks. And this question is saying, part A, question number 4A, is saying, explain how the following factors affect the speed of transmission of nerve impulse. The factors that affect the transmission. Explain how the following factors affect the transmission of nerve impulse. The first one is myelin sheath. Myelin sheath. Myelin sheath. Myelin sheath. Myelin sheath. How the myelin sheath affect the transmission of nerve impulse. How the myelin sheath affect the speed of transmission of nerve impulse. The myelin sheath is just a layer that acts as an insulator. Suppose that you have this as an axon. Then the myelin sheath are this insulator, which usually they are formed from Schwann cells. There's this nucleus somewhere here. Then the myelin sheath are formed from these Schwann cells. Then if this myelin sheath, like this, the points where there is no myelin sheath, like here, the myelin sheath is an insulator. This is the node of Ranvier. Node of Ranvier. Node of Ranvier. The myelin sheath affects the speed of transmission of nerve impulse because when the nerves, the axon is myelinated, means it has myelin sheath, it is wrapped by the myelin sheath, then the only following the life of a nerve impulse, suppose that this nerve impulse is being propagated in this direction, then only the portions where there is no myelin sheath are the ones that are permeable to charges. So following the life of this, that means if this region is where there is a nerve impulse, then this portion here becomes permeable to sodium. Therefore, if it becomes permeable to sodium, the sodium rushes in, and therefore the inside becomes because if the sodium rushes in, because when the cell is at rest, it's usually negative inside and positive outside when the cell is at rest. So the alive of this impulse is the one that triggers the permeability of this the membrane to sodium. So the sodium rushes in, from outside where it is in high concentration to the inside. So if this myelin sheath causes the charge to leap from one node of Langevin to another, this kind of movement is called saltatory movement. So the myelin sheath facilitates the saltatory conduction because the charges have to leap from one node of Langevin to another, because these are the only portions where the charges the charges can flow either in or out, because in all these other portions, these work as an insulator, so it does not allow the flow of charges either in or out. So this myelin sheath facilitates saltatory conduction. Saltere, which means to jump, jumping like movement from one node of Langevin to another, and therefore this increases the speed of transmission of nerve impulse. So axons with myelin sheath are said to be myelinated. Myelinated axons conducts the nerve impulse faster than the unmyelinated ones. Because for the unmyelinated ones, that means there is no myelin sheath, and therefore almost every portion becomes permeable to sodium, and therefore there will be the charges, because if this, suppose that this here becomes, this is positive, this is negative, and this is positive, this is negative, there will be very short circuits, many localized electrical circuits, which with this, then this one slows down the conduction of nerve impulse, because every portion of the axon is permeable to sodium. So that's how the myelin sheath affects the transmission speed of the nerve impulse. The second is temperature. Temperature, on average, as the temperature increases, also the speed of conduction of nerve impulse increases, because the speed of transmission of nerve impulse depends on the body temperature of the organism. And in this case, the transmission speed in homotherms is far greater than the transmission speed in the poikilotherms, because in most cases the temperature of the homotherms is higher than those of poikilotherms. In homotherms, the homotherms are capable of keeping their body temperature constant regardless of the fluctuations in the environmental temperature, and therefore on average they will have high temperature. The higher the temperature, the higher the transmission speed. The third factor that affects the transmission speed of nerve impulse is diameter of the axon, and this is very important. Diameter of the axon. The greater the diameter of the axon, the faster the speed of transmission of nerve impulse. Why? Because the diameter of the axon is inversely proportional to resistance. Is inversely proportional to resistance of the axoplasm. The larger the diameter, the lower the resistance. That means if you have an axon a big axon like this, and we have the ones which have small diameter, the resistance of the axoplasm in this is very low. The diameter is inversely proportional to resistance. As the diameter increases, the resistance of the axoplasm decreases, and therefore that allows the faster transmission of nerve impulse. This, if we are to explain this, don't forget this relationship. Diameter is inversely proportional to resistance of the diameter of the axon is inversely proportional to resistance of the axoplasm. Therefore, the larger the diameter, the lower the resistance of the axoplasm, and this allows will result into higher transmission speed of the nerve impulse. So this is because increased axon diameter minimizes the resistance of the axoplasm. Diameter is inversely proportional to resistance. That's all about part A of question number 4. Then part B of this question, part B of the question is briefly describe how the following are achieved at the synapse. How the following are achieved at the synapse. Briefly describe how the following are achieved at the synapse. How the following are achieved at the synapse. I think we take a break, then I'll be back after some time. All right. I have said that the second question of part B of question number 4 is saying briefly describe how the following are achieved at the synapse. The first one is facilitation. Briefly describe how the following are achieved at the synapse. First, let's consider this structure of a synapse with maybe this as a presynaptic knob. Then we have this with something like this. These are receptor molecules and on the postsynaptic knob. This, because this makes up this, the postsynaptic knob, something like this. Then let's take this as an example of a simple diagram showing the synapse. So the question says, briefly describe how the following are achieved at the synapse. The first one is facilitation. How facilitation is achieved at the synapse. How facilitation is achieved at the synapse. This is number 4, is question number 4b, and Roman 1 is how facilitation is achieved at the synapse. This is a synapse. The nerve impulse, the nerve impulse, once it reaches here, you know the mechanism of how the nerve impulse pass to the postsynaptic, the transmission of nerve impulse. I'm not going to go into the detail of the transmission of nerve impulse across the synapse, but I want to make myself clear about facilitation. Facilitation involves, because one nerve impulse reaches here, then it passes to the postsynaptic knob and then it continues. But facilitation involves each single nerve impulse leaving, each single stimulus leaving the synapse more responsive to the next stimulus. That means the sensitivity of the synapse is increased, because once the stimulus, which in this case it will be in form of nerve impulse, once it has been propagated to the postsynaptic knob and continues, then the synapse is left more responsive. This involves leaving the synapse more responsive, leaving the synapse more responsive to the next stimulus. It involves leaving leaving the synapse more responsive to the next stimulus. Because once a single nerve impulse once a single stimulus has been propagated, then facilitation, the synapse, is one among the role of the synapse. Facilitation means that this place, this gap, the synapse is left more responsive to the next stimulus, and therefore this increases the sensitivity of the postsynaptic membrane, because once a single stimulus has passed here, that means this portion is left more responsive for the next stimulus, and that is how the facilitation is achieved at the synapse. That is how facilitation is achieved at the synapse. The next is how, because the question is saying how the following are achieved at the synapse. I've said the next is amplification. How amplification? Usually, the arrival of the stimulus here triggers the permeability of this to calcium, which then the calcium, in the increase in concentration, the arrival of the stimulus at the presynaptic node here triggers the permeability of this to calcium, which then calcium rushes into the presynaptic node. The concentration of calcium is the one that triggers the fusion of the vesicles, the synaptic vesicles with the presynaptic membrane, which later on the vesicles bursts, releasing the transmitter substance. And in this case, maybe let's say it's acetylcholine, which let's assume that this is a cholinergic neuron. Then the transmitter is acetyl. So the release of transmitter substance in the synaptic cleft is the one that which is recognized by the large plasma proteins. These are receptor. They recognize the structure of the acetylcholine, and therefore opens the sodium and potassium channels. Therefore, this triggers the opening of the channel, causes the sodium to rush in, and therefore forming an action potential in the postsynaptic. But amplification means that enough or sufficient amount of transmitter substances are released in the synaptic cleft, which this one, sufficient amount of transmitter substances are released at the synaptic cleft, which this one amplifies the incoming stimuli. So the coming stimulus, even if it's very weak, then it can cause excitation on the postsynaptic membrane, because sufficient acetylcholine are released at the neuromuscular junction or at the cleft by each impulse to excite the postsynaptic membrane to produce a response in the muscle fiber. That means a stimulus arriving at this place, at this synaptic cleft, even if it's very weak, then it can cause action potential in the postsynaptic neuron because of the enough or sufficient amount of transmitter substances that are usually released here. Yeah, this helps to amplify. That means the stimuli arriving at the synaptic cleft, however weak, are enough to produce an action potential in the postsynaptic neuron. The other is the inhibition, how inhibition is achieved at the synaptic cleft. Inhibition in the transmission of nerve impulse across the synapse. Sometimes the transmission may be prevented from passing to the postsynaptic membrane by the activity of certain transmitter or drugs. Sometimes some drugs prevent the passage of the opening of the sodium, and therefore there will be no propagation of nerve impulse to the next neuron. This is inhibition. It is inhibition because it inhibits. If the nerve impulse is inhibited at the synapse, there will be no response on the effectors. The last part of this question is part C, and this part C is about the differences between rods and cones. Differences between rods and cones. Rods and cones are the photosensitive pigments found on in the retina of the eye. The rods and cones, they have many or different differences, and some of their differences are anatomical. For example, the rods. In rods, the outer surface is rod-shaped, but for the cones, the outer surface is cone-shaped. That must be the first difference between them. The other differences includes the rods. They are more numerous than cones, usually 120 millions approximately rods, but the cones are fewer than rods, approximately 6 millions. The rods are distributed uniformly throughout the retina except at the fovea. The rods are uniformly distributed throughout the retina except at the fovea. But the cones, these cones are highly concentrated at the fovea. The rods have low visual acuity. Why? The rods, acuity is the sharpness of the image that we see. Now in the rods, the rods have low visual acuity because of the arrangement. 3 rods are connected to one ganglia and one bipolar neuron. That's, of course, for the rods, it increases sensitivity but lowers the acuity. So the rods have low visual acuity, but the cones have high visual acuity. The rods have high visual sensitivity because of the arrangement. 3 bipolar, 3 rods connected to the connection, 3 rods to one bipolar neuron makes the rods to have a high visual sensitivity, but the cones have low visual sensitivity. The rods are sensitive to low light intensity, and therefore they are mostly used during the night, but the cones are sensitive to high light intensity. The rods also, as found on the retina, the rods contain one visual pigment, which is rhodopsin, and there is only one form of rhodopsin, but the cones have one visual pigment that exists in 3 forms, and therefore, because of this, the cones can discriminate color because there are 3 forms of iodopsin. That the cones have one visual pigment, which is iodopsin, and there are 3 forms of iodopsin, and therefore, because of this, the cones can discriminate color, but the rods cannot discriminate colors. Those are some differences between the rods and the cones. And that marks the end of question. That marks the end of question number 4. Let's get into the other question, which is question number 5. Question number 5 is found in cytology, and it has 10 marks like other section A questions. It has 10 marks. It consists of 2 parts. Question number 5A says, in what ways is water important to living organisms? Give 6 points. Importance of water importance of water to living organisms. The importance of water to living organisms. Water serves different purposes. One of them is water serves in metabolic activity. That is called metabolic role of water. Water is used in hydrolysis of many substances, such as hydrolysis of proteins into amino acid. Water is also a medium for all cellular metabolic processes. For instance, a raw material in photosynthesis. Water is important. Think of germination. It's important in germination. Water is a medium because many reactions takes place in the presence of water during even germination. Water also hydrolyzes the food during germination, but water saves because it causes imbibition during germination. It causes budging of the tester, which later on the imbibition forces causes the tester to burst, allowing the emergence of radicals and plumeri. Water facilitates diffusion of materials across surfaces as it dissolves many substances. Many materials diffuse more easier when they are in solution form. That means they are dissolved in water. Water also serves as a solvent. Water as a solvent, it transports various substances from one part of the body to another. Water is part of the blood. Water is a component of the blood, and therefore it transports different materials. It not only in the blood, even in other fluids, of which these fluids serves as transporting medium. So water transports different substances, including the end products of digestion of food and even some metabolic wastes from one part to another. Water removes the I've already said, wastes in animals such as urea and even carbon dioxide. They are transported using water. They are transported in solution form, or if it's urea, it's transported by the blood from where it is formed in the liver to other parts, to the part where it is excreted, which is in the kidney. So that's water as a solvent. Water also as a lubricant. Water in animals such as annelids and nematodes, water exerts a hydrostatic pressure which helps to support and maintain their structure. This is known as hydroskeleton. It's called the hydrostatic skeleton. These organisms, such as the annelids, the earthworms, and the nematodes, they have this hydrostatic skeleton, which they use it as a support of their body. Water also is important in the osmotic influx of water in the plant cells generate the turbo pressure. We know the plant cells are capable, they have fixed shape because the influx of water into their cells generates turbo pressure, which supports herbaceous, the non-wood plants. The turbo pressure is also supports the primary. It supports the primary growth in woody plants. The turbo pressure supports the primary growth in plants, because it results into elongation of the plants. Because when you speak of primary growth, you're talking of the growth that results into an increase in length. Yes, so the water results. The water causes the vacuolar function, which forms the large vacuoles, and as the water enters, then the large vacuole budges, which exerts a pressure. I think it's called wall pressure, and this results into an increase if it is in the meristematic cells, it results into extension of the cells, which is, in this case, it results into an increase in length, and it is primary growth in plants. Water also, water as a lubricant, it makes various lubricating fluids in the body. For example, mucus, which supports the mucus which supports, we are saying as a lubricant, it aids in the movement. It aids in the movement. It aids in the movement in animals. The mucus which aids in the movement in animals such as snail, synovial fluid in joints of vertebrates, the pleural fluid in the lungs, because it's the inhalation and exhalation involves the antagonistic actions of the intercostal muscles, which when they during these actions, then the water, the pleural fluid, which contains water, lubricates and therefore prevents, reduces the friction between these parts. So due to its viscosity property, water takes part in lubricating body parts, and this allows the body parts to slide one another without any damage. Water, supporting roles of water, or water as a supporting medium. We have already said that in animals such as annelids and nematodes, water exerts a hydrostatic pressure which helps this to support and maintain their structure. This is known as the hydrostatic skeleton. Osmotic influx of water in the plant cells generates turbo pressure, which supports the plants, makes the non-wood plant to stand upright, to grow upright, but also it facilitates the primary growth in the wood plants. Water found in the humus, like the vitreous humus, like vitreous humor and aqueous humor of the eye. This water is important because it helps to maintain the shape of the eye. Also, the mammalian fetus is supported and protected by the amniotic fluid, which in this is largely water. The amniotic fluid is largely water because the water is a component of the amniotic fluid which supports and protects the mammalian fetus in the womb. Water provides habitat to some aquatic organisms such as fish. Water is a home for some organisms such as fish. The fish they spend their lives in water. They are adapted for they survive, and therefore water is a home for some organisms such as fish. Water also control body temperature in animals. In animals like like example the homotherms, they can control their body temperature regardless of the fluctuation in the environmental temperature. Therefore, one among the physiological process that they use when the environment is overheated is evaporation, that is sweating. Sweating has cooling effect. Of course, it allows water to be lost, but the water that is lost has cooling effect. Water is also an agent of seed dispersal of seeds, that water aids in dispersing some seeds. Also, water saves in the dispersal of larvae. Water is important in the movement of male gametes of animals, such as the sperm swim towards the fallopian tube in the fluid which also contain water. And even the lower plants, the bryophytes and the pteridophytes or the fern plant, they produce flagellated male sperms from the antheridium, and these flagellated male sperms swim towards their chagonia in the presence of water. The lower plants depend on water for their sexual reproduction because they produce flagellated male sperms that swim in the presence of water to the achagonia, and therefore the sexual reproduction in the bryophytes and pteridophytes depends on water. Yes, part B of this question is saying why is the cytoplasm an important part of the plant? Give 4 points. Why is the cytoplasm an important part in plants? This question needs the function of the cytoplasm. The cytoplasm is a ground, jelly-like substance containing different substances within the cell. It is where the other cell organelles are suspended. The cytoplasm performs the following functions. First, it provides medium for the chemical reactions such as glycolysis. Glycolysis at the first stage of respiration takes place in the cytoplasm. So the cytoplasm is where the cytoplasm is the site for the some important chemical reactions or some important metabolic processes such as glycolysis. Glycolysis takes place in the cytoplasm. Glycolysis takes place in the cytoplasm. So it is a site for different chemical reactions such as glycolysis. It stores waste materials such as nitrogenous waste. The cytoplasm stores different waste materials such as nitrogenous waste. The cytoplasm also stores useful materials such as starch, glycogen, and lipids. It stores useful materials such as starch, glycogen, and lipids. The cytoplasm also aids in the movement of materials because it allows the material to move from one organelle to another. The cytoplasm is the site where other organelles are suspended. Also, the cytoplasm harbors and organizes the cell organelle that performs different functions such as protein synthesis in ribosomes and lipid synthesis in the smooth endoplasmic reticulum. That marks the end of question number 5. So question number 5 is of cytology with 10 marks. Cytology with 10 marks. And first part of this question is of the laws of water. That means it's cytology 2 in the chemicals of life, which is biochemistry, and part B is of the cell organelles. That means it's cytology 1. Question number 6 is of nutrition. This question also contains 6. It has 10 marks. It's divided into 4 parts that are labeled A, B, C, and D. And the first question is, I've said, is of nutrition. And part 1 is nutrition in plants, because the question says, distinguish between photoautotrophic from chemoautotrophic organisms. Distinguish between photoautotrophic from chemoautotrophic organisms. Then the photoautotrophic organisms are the organisms that use light energy from the sun as energy source for synthesizing organic compounds from inorganic substances with carbon dioxide as a source of carbon. These photoautotrophics, the photoautotrophic organisms includes the plants, because plants make their own food. They make their own organic molecules by using energy from the sun and carbon dioxide as a source of carbon. So the photoautotrophic organisms use light energy from the sun as a source, as energy source for synthesizing organic compounds from inorganic materials with carbon dioxide as a source of carbon. While the chemoautotrophic organisms, these use organic compounds which are synthesized from pre-existing organic sources. These use organic compounds which are synthesized from pre-existing sources of carbon using energy obtained from chemical reactions. That's the basic difference between photoautotrophic organisms and chemoheterotrophic organisms. The second question of the second part of this question number 6 is, by using 3 points, contrast cyclic from non-cyclic phosphorylation. Cyclic from non-cyclic phosphorylation. The differences between cyclic and non-cyclic phosphorylation. Cyclic, contrast, differentiating the cyclic from non-cyclic phosphorylation. I think the first must be the pathway of electrons. That in the cyclic phosphorylation, the electrons return back to its original state. That means the pathway of electron is cyclic. because the electrons are excited and then they return back to the original position. But in the non-cyclic phosphorylation, the pathway of electron is non-cyclic, is not cyclic. Why? Because the excited electrons do not return back to the original state. Instead, they end in the formation of reduced nicotinamide adenine dinucleotide phosphate, which is forms. That means it results into the excited electrons being trapped in the NAD. The other difference between the two is the cyclic phosphorylation. The first electron donor is photosystem 1. In the cyclic phosphorylation, the first electron donor is photosystem 1, abbreviated as P700. But in the non-cyclic phosphorylation, the first electron donor is water. The first electron donor is water. Again, the last electron donor in the cyclic phosphorylation, the last electron donor is also photosystem 1. But the last electron donor in the non-cyclic is NADP, because the electrons ends at NADP, because they are taken and the NADP becomes reduced to NADPH plus H positive. The products in the cyclic phosphorylation, the products are only useful ATP, but in the non-cyclic phosphorylation, the products includes the useful ATP and reduced NADP, but also there is waste water. There is waste oxygen, waste oxygen, which is a waste product in this non-cyclic phosphorylation. The photosystems involved in the cyclic only, it involves photosystem 1, but the non-cyclic involves photosystem 1 and 2. So that is question number 6B. Question number 6C is from nutrition. Again, it's still nutrition, but this time is nutrition in animals, because the question is saying, why is it necessary for pepsin to be secreted in an inactive state as pepsinogen? That means, why is pepsin secreted in an inactive state, which in most cases as pepsinogen? Then the answer is simple. Because the active pepsin would digest cells that produces it. There being no mucus barrier within the gastric gland, because in the gastric gland there is no mucus. Therefore, if the pepsin is secreted in an active form, then the pepsin would digest the cells that produces it, simply because in the gastric gland there is no mucus. The reason as to why the pepsinogen is secreted the pepsin is secreted as pepsinogen, as inactive form, is to avoid the pepsin, is to make it, because if it is inactive, then it prevents, it does not digest, it does not destroy the cells producing it. And part D of this question, which is the last part of question number 6, is saying, list 3 features of the small intestine which increases its surface area. The small intestine, the part of the small intestine which is responsible for the absorption of any products of digestion of food, is the ileum. The ileum has some features that increases its surface area for the absorption of any product of digestion. The first feature is the lining or the wall of the ileum. The wall of the ileum is highly folded. The wall of the ileum is highly folded. the wall is folded in order, it has the wall of the irium is highly folded. If it's folded, that means it increases, it maximizes the time for the absorption of food. The wall of the lining of the irium is highly folded, which increases the surface area for absorption of food. Again, the lining, the irium, it has the finger-like projections called villi. These villi are these ones. Villi are like these ones, which increases the surface area for the absorption of the end products of digestion. The villi are further divided into microvilli, which also the subdivisions of the villi, the microvilli are smaller than the villi, and they increase the surface area for absorption of end products of digestion. The absorption of end products of digestion. And that marks the end of question number 6. The next question is question number 7, which this question number 7 is the last question in section A. Last question in section A. This question is saying, part A, question number 7A. Explain the features which are found in both prokaryotic and eukaryotic cells. Give 2 points. That means this question is about the similarities between a eukaryotic cell and a prokaryotic cell. So the eukaryotic cell and prokaryotic cell possess the following similar feature. Both have the following features. 1, they have cell membrane that separates the cell's interior from the surrounding environment. That all the cells have the cell membrane, both prokaryotic and eukaryotic cells have cell membrane that separates the interior part of the cell from the surrounding environment. They also both have a cytoplasm that consists of a jelly-like region within the cell in which all other cell organelles are floating or suspended in the cytoplasm. And then also they both have ribosomes. They have these ribosome particles that synthesize protein and genetic material of the cell. Those are the 3 similarities between prokaryotic cell and eukaryotic cell. Part B of this question, of question number 7, is saying, briefly describe the following interaction or bonds in the formation of protein. The first one is called the hydrogen bond. Hydrogen bond. The meaning of hydrogen bond. Hydrogen bond because describe the following interactions or bonds in the formation of protein. In the proteins, hydrogen bonds is a bond. Any hydrogen bond in protein is a bond formed in the polypeptide chain between amino acid and side groups. It's a bond formed between amino acid in the polypeptide chain between amino acid and the side group. The bond is an electrostatic attraction between hydrogen atom and another electronegative atom, such as nitrogen and oxygen. So it is an electrostatic attraction between hydrogen and another electronegative atom, such as nitrogen and oxygen. The hydrogen bond, if you have, suppose that you have this protein, that you have amino acid, not protein. If you have, say, hydrogen bonded to nitrogen, this nitrogen has this, then there is C with the R group and hydrogen. There is this one here. Yes, this is a simple amino acid. Then when this simple amino acid, for example, reacts with another of the same, that is N with hydrogen, here there is this with this one, and then this with this double bond OH. The reaction must be condensation. Suppose that this combines with, suppose that this one here combines with this, so it forms nitrogen with hydrogen bonded to this with hydrogen. There's R, this here is this, then this is bonded to nitrogen with the hydrogen, then something like this. Here is R, the carboxylic group, here like this. Then the hydrogen bond forms between carbonyl, that is between this and this. So the bond between this one here, this weak attraction between this one, is hydrogen bonding. Or it can be, maybe, if it can either be maybe a bond between it can be a bond of this oxygen and hydrogen from another amino group of another amino acid, of which this here is hydrogen. A bond between hydrogen and another electronegative element such as nitrogen and oxygen in this context, because it's the hydrogen bond in the formation of amino, in the formation of polypeptide chain, or in the formation of protein. The second type of bond is ionic bond. Ionic bond occurs between the positively and negatively charged side chains that will come into contact with each other. It occurs between the positively, and usually is the ionic bond in the protein is the bond between this one here. It's the bond between this and this one here. The bond between this and this. The bond between this one here and NH3, this one, is called ionic bond in the formation of protein. So it is the bond that occurs between acidic group and basic group of the amino acid. It occurs between positively and negatively charged sides, that of side of the chains that comes into contact. And the last in the formation of protein is the peptide bond. Peptide bond is the bond between the acidic carboxylic group, this one, the acidic carboxylic group, and the basic amino group. For example, here, then this is peptide linkage. It's peptide bond or linkage. This is peptide bond or linkage. So it is, the peptide bond is a bond between carboxylic group of one amino acid and the basic amino group of another amino acid. So it's a bond formed between the carboxylic group of one amino acid and the basic amino group of another amino acid. It's the bond between this group and this, which results into this one. Of course, this is a condensation, right? Because there is formation of water molecule. It's a condensation reaction where the formation of polypeptide showing the formation of peptide bond forms water, and therefore it's a condensation, and it's a bond between the carboxyl group and the basic amino group. The polypeptide molecule formed when during the formation of peptide linkage, the peptide bond results in formation of a molecule with a free amino group on one side and free carboxyl group on the other side, or at the end of each one. For example, in that one, if there is a free amino group at that end, on the right hand end, and there is formation of there is a free carboxyl group. There is a free amino group at this end and a free carboxyl group at this end. Yes, that marks the end of the section A of paper 1 with 7 questions. It has 7 questions, each with 10 marks. Yes, let's get into the second section of this biology 1, and there are some questions. There are some review questions that you can use to revise. These review questions are there are 3 questions of section B. Question number 8 is a question of transport of materials in living things. Part A is saying when an animal is wounded, its overall blood pressure rises, but the area in the vicinity of the wound swells as a result of local vasodilatations. Suggest what might be the advantages of these changes. When an animal is wounded, its overall blood pressure rises, but the area in the vicinity of the wound swells as a result of local vasodilation. Suggest what might be the advantages of these changes. If you're given this question, the possible answers of this question are the local vasodilation in the wounded area enables more blood to flow, enables more blood carrying oxygen and nutrients to arrive at the wounded part. And this is important because it speeds up the process of repair and replacement. So the increase in pressure in the local vasodilation in the wounded area enables the blood carrying the nutrients and oxygen to arrive at that place and speed up the process of healing or the repair and replacement. And also the increased body blood pressure prepares the body of the animal to respond to any further stress more readily and efficiently. Part B of this question is saying, also of transport, is saying outline the main adjustments that occur to the heart rate and circulatory system just before, during, and after a 10-meter race. Outline the main adjustment that occur to the heart rate and circulatory system just before, during, and after a 10-meter race. The adjustments are divided into 3. Before the race, before a 1-meter race or before a 1-meter run, first, adrenaline hormone is secreted in anticipation of the race. But again, this stimulates vasoconstriction throughout the body in all but most vital organs. Hence, blood pressure is raised and also heart rate is also increased. During the race, there is an increase in metabolic activity because of adrenaline. Increased metabolic activity takes place during the race, especially in the skeletal muscle. This increased metabolic activity means there will be production of more carbon dioxide, because from this increased metabolic activity, one among the fundamental metabolic activities is respiration, of which respiration produces carbon dioxide. So the increased metabolic activity during the race, especially in the skeletal muscle, results into an increased carbon dioxide levels in the regions which in different parts of the body, and this promotes local vasodilation because of high partial pressure of carbon dioxide. The increased body also metabolism increases the temperature, or metabolism generates heat from within. Therefore, the increased body temperature further enhances vasodilation. However, the general increase in carbon dioxide level in the blood is noted by the chemoreceptors of the aorta and carotid bodies, which in turn stimulate the vasomotor center to promote vasoconstriction. This vasoconstriction, this increases the blood pressure and speeds up the blood flow because of vasoconstriction. Heart rate is also increased and more complete emptying of the ventricles occur. Towards the end of the race, the muscles will be respiring anaerobically and producing large amount of lactic acid. Then strong contractions of the muscle squeezes the veins and promote fast venous return to the heart. The third phase is the recovery phase, where in the recovery phase, oxygen debit is paid off and lactic acid is removed from the bloodstream. By paying the oxygen debit and the removal of lactic acid, that means the lactic acid is being converted, is being transported from the muscles back to the liver so that it is converted back to pyruvate, because during the race there was an aerobic respiration that produced lactic acid. Also, the tissues subside in activity and the carbon dioxide levels decreases, and consequently there is a return to normal heartbeat and hence normal blood pressure. That is the end of question number 8. Question number 9 is of reproduction. Question number 8 is of transport of materials in living things, and it has 15 questions. As usual, according to the NECTA format, a Form 6 in the Form 6 national exam will have Section A and B. Section A with Biology 1, Section A with 7 questions, and they are supposed to answer all questions, and each one has 10 marks. Each question has 10 marks, therefore Section A makes up 70, it has 70 marks, and Section B with 3 questions where the students will be required to opt for any 2 questions. The first, the one that I have said, is a question of transport of materials in living things with 15 marks. And the other question that the students can opt is this question of reproduction. Question number 9. This question is saying, describe the hormonal control of oogenesis. Oogenesis is the formation of eggs. Oogenesis is the formation of eggs in mammals. The formation of eggs in mammals is called oogenesis. The formation of eggs in female mammals is called oogenesis. The control centers for the production of eggs in mammals are the hypothalamus and pituitary gland working together. The hypothalamus secretes the gonadotrophin releasing hormone. The hypothalamus secretes the gonadotrophin releasing hormone. Suppose that this is the hypothalamus. The hypothalamus secretes the gonadotrophin releasing hormone. Hypothalamus secretes, I've said, the formation of eggs is controlled by both the hypothalamus and pituitary gland working together. The hypothalamus secretes gonadotropin releasing hormone, abbreviated as this GnRH, which stimulates the pituitary gland, and in this case, the anterior pituitary gland. This stimulates the anterior pituitary gland. This stimulates the anterior. The hypothalamus secretes the gonadotrophin releasing hormone, which the gonadotrophin releasing hormone stimulates the anterior pituitary gland to secrete the hypothalamus secretes gonadotrophin releasing hormone, which stimulates the anterior pituitary gland to secrete the hormone called the FSH, follicle stimulating hormone. This stimulates this anterior. That means the hypothalamus secretes gonadotrophin releasing hormone, which stimulates the anterior pituitary gland to secrete follicle stimulating hormone, FSH. Follicle stimulating hormone travels in the blood to its target, and here the target of the FSH is the ovary. In the ovaries, FSH feeds into receptor sites in the primordial follicles. In this case, the FSH stimulates the development of several follicles, but only one of which will complete the development per one cycle. As the FSH causes the development of the follicles, then the outer layer of the follicle, called the granulosa cell, of the granulosa cell of the developing follicle, starts to produce a female sex hormone. This starts to secrete a female sex hormone called the oestrogen. Oestrogen is a steroid hormone, and this hormone has two targets. The oestrogen has two targets. The first target of oestrogen is the anterior pituitary gland, and the second target of the oestrogen is the uterus. I repeat again, this oestrogen that is produced by the layer, the granulosa cell of the developing follicles. The oestrogen is a steroid hormone, and it has two targets. The first target is the anterior pituitary gland, and the second target is the uterus. In the anterior pituitary gland, the oestrogen switches off the secretion of FSH. In the anterior pituitary gland, the oestrogen switches off. The oestrogen inhibits the secretion of FSH in the anterior pituitary gland, and therefore it prevents the possibility of further follicles being stimulated, so that only one egg is produced at a time. In the second target, which is in the uterus, the oestrogen stimulates repair and development of the lining of the uterus, called the endometrium. This is in preparation for the possibility of pregnancy when the embryo will implant in the endometrium. At the midpoint, when there is high level of oestrogen triggers the secretion of luteinizing hormone from the anterior pituitary gland. The high level of oestrogen triggers the secretion of luteinizing hormone from the anterior pituitary gland. The luteinizing hormone and follicle stimulating hormone are released in a surge. The surge ensures precise timing of ovulation. Luteinizing hormone, the target of the luteinizing hormone is the ovary, where luteinizing hormone in the ovary causes ovulation. This here causes ovulation. Ovulation is the release of secondary oocyte when the mature Graafian follicles bursts. So the luteinizing hormone induces ovulation in the ovary, but also the luteinizing hormone converts the remains of the Graafian follicles after the Graafian follicles have busted. It converts the remnants of the Graafian follicles into a yellow body. This yellow body is called the corpus luteum. Corpus luteum. The corpus luteum is an endocrine structure. The corpus luteum is an endocrine structure, especially in the first 3 months of pregnancy, before the placenta is fully formed. The corpus luteum secretes large amount of progesterone and small amount of estrogen. Like large amount of progesterone and small amount of estrogen. Like estrogen, progesterone is also a steroid hormone, and it has 2 targets also. The first target is the anterior pituitary gland, and the second target is the uterus. Where in the uterus, progesterone stimulates the uterus to maintain its thickening, and also stimulates the glandular activity. In the anterior pituitary gland, progesterone switches off the secretion of luteinizing hormone. In the anterior pituitary gland, it inhibits the release of luteinizing hormone. The release of progesterone is associated with a rise in body temperature of the female just after ovulation. If fertilization does not occur, the corpus luteum starts to disintegrate after 28 days in the cycle. The level of estrogen and progesterone therefore decline, so the inhibition of FSH and luteinizing hormone is removed. Therefore, if there is no progesterone, the endometrium breaks down, causing menstruation in primates. So the disintegration of the corpus luteum the disintegration of the endometrium together with the secondary oocyte comes out together with the blood vessels, and the material discharged are called the menses or menstrual fluid. That is marks the end of the first. That marks the end of part A of question number 9. Part B of question number 9 is about the differences. The question want the students to write the differences between complete and incomplete metamorphosis. Complete metamorphosis, it involves 4 stages, which are egg, larva, pupa, and adult. But the incomplete metamorphosis involves only 3 stages, which are egg, nymph, and adult. The complete metamorphosis, larva and pupa are present, and larva is a feeding stage, while the pupa is a non-feeding stage. But the larva is different from the adult because the larva consists of the mandibles for piecing or tearing the leaves of the trees, while the adult in the complete metamorphosis feeds on, they have proboscis for the usually sucking nectaries from the flowers. But in the incomplete metamorphosis, larva and pupa are missing. In the complete metamorphosis, there is no nymph stage, but the complete metamorphosis, in complete metamorphosis, there is nymph stage. So complete metamorphosis, for example, complete metamorphosis, this is also called holometabolous. Holometabolous, where the egg hatches into larva, which these are the larva, then pupa, the pupa is adult. The adult lay the eggs and the cycle begins again. This is complete metamorphosis, or otherwise called the holometabolous. But in complete metamorphosis, consists of 3 stages. This is also called the hemimetabolous. It has 3 stages, which are egg, then nymph, then adult, where the adult lay eggs and the cycle begins again. So complete metamorphosis involves 4 stages, but incomplete involves 3 stages. Complete metamorphosis consists of larva and pupa stages, but incomplete lacks the larva and pupa stages. Complete metamorphosis, there is no nymph stage, but there is nymph stage in the incomplete. The nymph resembles the adult, but the larva, which is the second stage, is different from the adult. Examples of organisms here, I think this includes, example of organisms in this include butterflies, undergoing complete butterflies, even houseflies. But this here, the examples of this includes cockroaches, and even grasshoppers. These undergoes incomplete metamorphosis. That marks the end of question number 9. Let's get into the last question, which is question number 10. Question number 10 is a question of respiration, and the question is saying aerobic respiration involves, number 10A, aerobic respiration involves glycolysis, Krebs cycle, and the electron transport system. Describe the importance of the Krebs cycle. The Krebs cycle is the second stage of aerobic respiration. The Krebs cycle is important because of the following. First, the Krebs cycle brings about the degradation of macromolecules. That means it's a means by which the large molecules are broken down into simpler molecules. For example, pyruvate, which is CH3, C double bond H. This is the pyruvate. This is converted to carbon dioxide. this is degradation of, shows that pyruvate is a 3-carbon compound, is converted into moles of carbon dioxide. So it brings about the degradation of macromolecule into simpler molecules, like the conversion of glucose, which is a 6-carbon molecule, into pyruvate, but also the conversion of pyruvate to carbon dioxide. It also provides the reducing power for the electron transport system. In the Krebs cycle, from the Krebs cycle, 1 ton of the cycle, 1 ton of the Krebs cycle, 1 ton of the Krebs cycle produces pairs of hydrogen. For example, 1 ton of the Krebs cycle produces 3 reduced NAD, NADH plus H positive. But also it produces 1 ton, 1 FAD H plus H positive. This is per 1 ton, per 1 cycle. That means from 1 glucose molecule, this will be times 2 for 1 glucose. So it means it is 6 NAD H2, and also here it is 2 FAD H2. So these are the raw materials for the electron transporting system. These are the ones that enter into the ETC, because they are the raw materials for the electron transporting system. So it produces the reducing power for the electron transport system, as it produces pairs of hydrogen atoms, which are ultimately the source of metabolic energy for the cell. It is also an interconversion center. The Krebs cycle is an interconversion center because it is a valuable source of intermediate compounds as used in the manufacture of other substances. It produces materials such as the alpha-ketoglutarate, the citrate, fumarate, malate, succinate, of which these can be used in the synthesis of other substances such as fatty acids, amino acids, and even chlorophyll. The Krebs cycle also produces fumarate, which the fumarate, the fumarate can be used in the synthesis of other substances, as it is also, there is fumarate that comes from the ornithine cycle in the Krebs bicycle, where the fumarate from the ornithine cycle can enter the Krebs cycle. So it allows it uses some other materials produced by the cells to produce energy. Therefore, it is an interconversion center. That is question number 1. Number 10A. Number 10B. Question number 10B. Outline how lipids is maneuvered as a respiratory substrate. Outline how lipid is maneuvered as a respiratory substrate. Lipids as respiratory substrates. Lipids as respiratory substrate can be maneuvered and can be converted into the Krebs cycle. Can be maneuvered into forms so that they can join the respiratory pathway of glucose. For example, first the lipids are usually hydrolyzed into their component, their monomers, which are fatty acid and glycerol. Then the glycerol is phosphorylated and converted into triose sugar, which is then converted into pyruvate. Then it enters the ring stage to form acetyl, where the acetyl is carried by the coenzyme A to form acetyl coenzyme A, which this enters the Krebs cycle in the matrix of the mitochondria. The other component, which is fatty acid. The fatty acids are first broken down into fragments, each fragment with 2 carbon atoms. So the number of fragments made depends on the number of carbon atoms found in the fatty acid. Later on, each of these 2 carbon fragments is converted into acetyl, then carried by the coenzyme A, which then joins the Krebs cycle. If you are to show that the pathway in like this, if suppose that you are, maybe we get a break, so that when I will be back so that we finish at the part of this question. Yes, let's proceed with this last part, which we were on question number 10b, the respiratory pathway of lipids. I'll say that the lipids are first hydrolyzed. Suppose that you have this, maybe the glucose is converted to pyruvate. Pyruvate then is converted to acetyl, which is carried by coenzyme A, then enters the Krebs cycle. It first forms citrate, which is a 6-carbon compound, and here it combines with oxaloacetate, which is a 4-carbon compound. So here then it's converted to alpha ketoglutarate, this involves decarboxylation where carbon dioxide is lost, and also a single NAD positive picks a pair of hydrogen and becomes NADH2. Then this is a 5-carbon compound. This is further converted to succinate, which is a 4-carbon compound. Here there is formation of 1 ATP, or otherwise called GTP, and also this involves this decarboxylation, where carbon dioxide is lost and is used to form carbon dioxide, and another NAD, the reduced NAD picks a pair of hydrogen and becomes reduced. Then the succinate is converted to fumarate. Here is a pair of hydrogen, which here there is fat, and a single fat picks a pair of hydrogen and becomes FADH2. Here then is converted to malate, and this malate is a 4-carbon. This is a 4-carbon. This here involves hydration, where water, and then this is converted to malate here, where this another NAD that picks a pair of hydrogen and becomes NADH2. So if it is how the respiration of lipids, lipid is first hydrolyzed into 2 components. One is this is glycerol. That the glycerol is a 3-carbon is phosphorylated to form 3-PGAL, which then joins here. The lipid, the other component of this is fatty acid. Fatty acid is progressively broken down in the beta oxidation, and then to form 2 carbon fragments. Each of the fragments forms the acetyl, like is carried by the coenzyme A, and then it enters the pathway of glucose. That is question number 10. Number 10B. Number 10C is an equation. This equation shows that the equation below shows the relationship between various metabolic processes. Various metabolic processes. There is glucose, then process A. Here it forms pyruvate. This forms pyruvate. Then carbon dioxide and ethanol. This side is carbon dioxide and ethanol. Ethanol, yes. The other side is carbon dioxide and water. This way is carbon dioxide and water. The process, this is represented by letter B, and this is C. Yes. Identify the 3 metabolic processes represented by letters A, B, C. From this, if it results into pyruvate, then A must be glycolysis. A is glycolysis because it's the conversion of glucose to pyruvate. B is conversion of pyruvate to carbon dioxide and ethanol. Obvious, B is alcoholic fermentation. Alcoholic fermentation. That is anaerobic respiration in yeast. Anaerobic respiration in yeast. C is aerobic respiration. Aerobic respiration. Because it forms carbon dioxide and water. That's C Roman 1. C Roman 1. These are C Roman 1. C Roman 2. C Roman 2. State the specific site where each of the 3 metabolic processes occur. State A, glycolysis, takes place in the cytoplasm of the cell. This takes place in the cytoplasm of the cell. B takes place, this B takes place because it occurs also, it's alcoholic fermentation. It occurs in the cytoplasm of the yeast cell. Cytoplasm of the yeast cell. And C is, if it's aerobic, that means it shows the other 2. That means there is, this involves 2 stages because there is Krebs cycle, which occurs in the matrix of the mitochondria, and there is electron transport system, ETC, electron transport system that occurs on the crystal. Therefore, generally, this C occurs in the mitochondria. It occurs in the mitochondria. That is C Roman 2. C Roman 3 How many ATP molecules are formed at each of the stages A, B, C? A, only 2 ATP molecules are produced, glycolysis. B, no ATP, no formation of ATP. C is aerobic respiration. That means there is Krebs cycle that 2 ATP molecules are produced, and we have the electron transport system, ETS, where here 34 ATP molecules are produced. 34 ATP molecules are produced. Yes, then number 10D, glycolysis leads to formation of pyruvic acid. How is the pyruvic acid converted into ethanol? Explain by giving 2 points. Pyruvic acid, that's the conversion of pyruvate into ethanol. That is the anaerobic respiration in yeast. First, it involves 2 reactions. The first one is decarboxylation, where here in the decarboxylation pyruvate from glycolysis is converted to ethanol through the removal of carbon dioxide molecule. That is, you have CH3, this is pyruvate. The pyruvate, there is a bond, OH here. The pyruvate is converted to CH3, C double bond, this, which is ethanol, right? It's converted to ethanol, and this ethanol is an aldehyde. So here, carbon dioxide is produced. There's formation of carbon dioxide. Then later on, there is a reduction where ethanol is reduced. Ethanol combines with a pair of hydrogen from. So this is the first. The second stage is the ethanol, CH3, this double bond, hydrogen combines with the hydrogen, pair of hydrogen from this, and here it forms CH3, CH2, OH. This is ethanol. This is pyruvate. Here there is carbon. Pyruvate, and this is ethanol. So it involves 2 stages. Decarboxylation, where pyruvate is converted to ethanol, then carbon dioxide is evolved, then ethanol is converted to ethanol by accepting a pair of hydrogen to form ethanol. The ethanol is converted to ethanol. And that marks the end of our session. Thank you.